Let's take a look at the last phase of aerobic cellular respiration, and that's the electron transport chain. So this diagram should look a little bit familiar. It's very similar to the one that you saw in photosynthesis across the thylakoid membrane. And we have the matrix, which is that fluid inside the folded inner membrane. This is the inner membrane here, okay, so where all those folds happen. And this is the inner membrane space. So looking at how this starts, okay, NADH and FADH2 pass high energy electrons along the membrane proteins on the cristae. Now remember that the NADH and the FADH2 come from the previous reactions. So here we have our NADH and our FADH2. You can see that the NADH is going to give off those electrons and then becomes an acceptor again, and the FADH2 gives off the electrons and becomes FAD. So these are going to pass those high energy electrons along the membrane proteins, okay, which are these big round blue things, and you can see the electrons start bouncing. Remember from photosynthesis that we had our ground state electron, which had little energy, and when it's energized or it's given energy, it's going to start to bounce. So it starts to bounce along the membrane along these proteins. Now, the final electron acceptor in this process is actually oxygen. And oxygen is going to grab these hydrogen ions that are here. And hydrogen plus oxygen is going to equal water. So we're actually going to get two water molecules from this process. The next stage is actually going to show us that as the electrons move, their energy is going to pump out hydrogen ions by active transport. So in this matrix space, we have some hydrogen ions, and we're going to use some of the energy from the electrons bouncing to actually push those hydrogen ions back across the membrane into the intermembrane space. And when that happens, we're going to create a concentration gradient with all these hydrogen ions in the intermembrane space and very few in the matrix. And then because of that, you can see that these are all positive. The intermembrane space becomes positively charged, and the matrix is negatively charged. And the last part of this actually is with that concentration gradient, because we have so many hydrogen ions up here and we have a concentration gradient, they're going to want to move to where there are less ions. And they're going to move by the process of chemiosmosis, just like in photosynthesis. So as they move, the hydrogen ions are going to pass through the ATP synthase, and that's going to cause the ATP synthase to spin. And as that spins, the ADP is going to pick up those phosphates and become attached, so it forms ATP. We get lots of ATP from this process. So if we finish filling out our chart, you can see that the electron transport chain happens in the cristae, or those folded membranes, and we have the NADH and the FADH2 from glycolysis in the Krebs cycle to start with, and our products are ATP and water. We didn't use any ATP in this process, but we made 34 ATP, and our net gain is 34 ATP. So it gives us a total, after the electron transport chain is completed, somewhere between 36 and 38 ATP. It varies from time to time, and these are our general counts. So anywhere from 36 to 38 ATP total for cellular respiration aerobically. So if we look at an overview again, we start off with glucose. From there, okay, we go through the process of glycolysis, which happens in the cytoplasm. That glucose is broken down into pyruvic acid, and that pyruvic acid, if oxygen is present, is then going to go to the Krebs cycle, which happens in the matrix of the mitochondria, that liquid-filled space. From there, now we go to the electron transport chain, which happens in the cristae, or the folded membranes. So we start with glucose from foods we eat. Glycolysis happens, which is the anaerobic stage, forms pyruvic acid. That pyruvic acid, in the presence of oxygen, will move to the Krebs cycle, and then the products from the Krebs cycle also move to the electron transport chain. Now let's compare. I keep talking about photosynthesis, and let's look at cellular respiration and photosynthesis together. The purpose of cellular respiration is to make ATP from food, and photosynthesis' purpose is to make sugar from sunlight. Okay, so they're related here because we take that sugar and then make ATP. The chemical reactions that are involved in cellular respiration are glycolysis, the Krebs cycle, and the electron transport chain. In photosynthesis, we have two reactions, the light reaction and the Calvin cycle. The electron acceptors, we've talked about those. Those are what's going to pick up the extra electrons. We have oxygen in the electron transport chain is going to be the final electron acceptor. And then we have NAD plus and FAD. In photosynthesis, these electron acceptors are the NADP plus. Now, we know that cellular respiration happens in the cytoplasm with glycolysis, and the Krebs cycle happens actually in the mitochondria. And photosynthesis takes place in the chloroplast, and we divide that up. The light reactions happen in the thylakoid membranes, and the dark reactions happen in the stroma. We have equations in cellular respiration for glycolysis, the Krebs cycle, and the electron transport chain. And then we have our overall equation for photosynthesis with water and carbon dioxide yielding glucose and oxygen. In cellular respiration and photosynthesis, the types of transporter that used are both chemiosmosis and active transport. The chemiosmosis, remember, is going to be the hydrogen ions moving through this ATP synthase because of the concentration gradient that's created, and the active transport is going to be used to pump those hydrogen ions back into the intermembrane space in order to create the concentration gradient.